All right. So for today's final lecture, the content is going to be focused on cell cycle and how it relates to cancer, in particular cervical cancer. Um, the content for this lecture was originally 40 slides. Given that we have a class today where we took a quiz, I've tried to condense it down to about 25 slides. And so there's going to be some slides that we skip over just for the sake of time. And so the content that we cover is what's going to be uh, is what you'll be assessed on. You won't be held responsible for the slides that we've skipped over, just to try and create clarity there. Those are still there for you to try and better understand some of the different specific information for each cell cycle step. However, the information that we focus on today is what you'll be accountable for. Uh, so given that the application's cancer, you can appreciate that there's a really high mortality rate from cancer, and that currently, with it being the leading cause of death, there's 1 in 4 deaths that are attributed to the variety of different kinds of cancer that have been studied and identified clinically. It's been identified that approximately 1,500 people a day are dying from cancer. The total cost is about 180 billion, and the direct medical expenditures are at 60 billion. And these numbers are about 6 years old right now, so these numbers have likely gone up over time. Um, the in addition to cancer, one of our application points is going to be human papillomavirus, or HPV. Um, it's a specific strain of virus that's been attributed to some some things that you all have been probably exposed to or have some experience with, whether it's common warts on your skin, plantar warts. But that same virus, different strains of it exist, and some of those strains have been also connected to different genital malignancies, primarily cervical cancer, as well as head and neck cancers as well, oral papillomas. And so what's important to know is that these different strains can have different outcomes. And the way we typically describe that is that some of these strains, particularly the ones that are associated with warts on different parts of your skin, we describe them as benign tumors that emerge. Particularly for those, we'd usually describe them as lesions. And so benign typically means that they're not going to be invasive or spreading to different parts of the body usually from that one specific lesion. However, the strains that are associated with cervical cancer or head and neck cancers are going to be identified as being malignant tumors. The difference, again, with benign versus malignant is going to be primarily boiling down to how invasive that lesion is, how invasive that tumor is, and its ability from that original lesion to spread out to other tissues. What's important to note is that the infection rate of HPV is fairly high. There's a bit of a distribution across age. Primarily, usually about 27% of women overall will be presenting with some sort of infection with HPV. The incidence of infection is incredibly high, close to approximately about 100% of everyone's lifespan. So there is a likely bet that at some point over the course of your life, you will have been exposed to HPV, whether that exposure leads to a common wart or potentially some connection to cervical cancer or head and neck cancer will vary greatly. But the plug here is that the great thing is that there are vaccines against HPV that inhibit HPV's effect in leading to cervical cancer or head and neck cancer. What's important also to note is that there's the HPV strains that are associated with cervical cancer are considered necessary. They're an important part of the pathogenesis of cervical cancer, but they are not in and of themselves sufficient to cause it. Meaning that whenever you see an instance of cervical cancer, that patient will test positive for HPV. But in a study where you deliver HPV, that in and of itself is not sufficient to induce, sufficient to induce cervical cancer or for a tumor to grow on the cervix there. And so whenever we talk about taking a step back and just like thinking more broadly about when you see information uh, from either a research article or more importantly from a news article that tries to highlight some link between red dye and health outcomes or fluoride in water, for example, and negative health outcomes, it's important to think about how those things are correlative or causal. And when we think about whether it's causal, one of the questions we oftentimes think about is that, is this variable, whatever this is it necessary or sufficient to induce that health outcome? And so primarily think about this being the last lecture, and one of the things I want you guys to walk away with is the tools you have before you from what we've done this semester to try and assess information that's provided to you and how you determine the strength of that data, the strength of their claim. And so it's important to think about things being necessary and sufficient. So in this case, HPV is necessary for cervical cancer. However, it in and of itself is not sufficient to induce a tumor to emerge on the cervix there. And this doesn't just only affect women, but also is the incident rates of tumors that emerge, or cancer incidence rate that emerges in response to HPV is also particularly high among men when it comes to the head and neck or the oral papillomas that emerge as well. And so the ability for HPV to have a role in promoting cancer doesn't just apply to women, but applies to everyone across the board. There's about the incidence rates for cervical cancer specifically is about 8 in every 100,000 per year. There's currently about 250,000 patients that have cervical cancer currently, and the mortality rate is about 2.4 per 100,000. The median survival rate, meaning the average age at which a person survives with cervical cancer, is about 54 years old. Usually the diagnosis for cervical cancer involves either a pap smear or cervical imaging. And so with the cervical imaging we have here, there's this lighter region around the cervix that is the lesion that is the malignant tumor there. And so these are primarily the means by which cervical cancer is diagnosed. It's important for us to try and define some key terms when we talk about cancer biology, as well as both terms that you may have heard of before, as well as terms that you've heard of before that you may be familiar with, as well as terms that you've heard of but don't really understand how they might fit into the story of cancer biology. And so again, when we talk about benign tumors or benign lesions, they're typically harmless, and they are the opposite end of what we could consider malignant. So benign typically lacks the features of what defines a malignant tumor. And so for a malignant tumor, typically what's key and what characterizes malignancy is this ability for cells to lose their differentiation, lose their specialty, which is defined as anaplasia. This ability for them to invade and move and migrate into the surrounding tissue around that primary tumor. And the last key thing is their ability to undergo metastasis, their ability to spread to totally other tissues. And so usually when we think about the staging of cancer, stage 1, stage 2, stage 3, stage 4, the final stage is usually defined by metastasis, where tumors emerge in totally other tissues. As an example, typically with breast cancer, stage 4 breast cancer will present with malignancies, oftentimes in the brain or in bone tissue. And so the characterizations of anaplasia, invasion, and metastasis define what is a malignant tumor. And the absence of those qualities is what characterizes a benign tumor. And tumorigenesis describes the initiation or beginning of a malignant tumor, or the beginning of malignancy. And so when we think about anaplasia and invasion, again, going to breast cancer, and so this is a histology slide of normal breast tissue that stains for collagen 9 alpha 3, and that's one of the key markers for triple negative breast cancer. And this is what it looks like typically in normal mammary tissue. But when we look at a slide of triple negative breast cancer, we can actually see that there's an invasion, this invasion to the surrounding tissue this loss of, we have here in the normal breast tissue, this epithelial gland where there's a lining of epithelium with collagen 9 standing all around it. But during malignancy, these epithelium lose their specialization where they've been oriented to create these cell-to-cell junctions. They have their polarity in apical side and a basal side, where the basement membrane is on their basal side. But during anaplasia, they lose that orientation, they lose their specialty, and the tissue totally alters their structure and their function that enables both invasion and eventually metastasis. And so collagen 9 alpha 3 or col9A3 is one of the genetic markers for triple negative breast cancer. Some additional cancer terms that are important to understand. When it comes to tissue, there's a critical need for oxygen to be present for those cells to survive. And typically there's kind of this oxygen diffusion distance where they need to be within a couple millimeters of some oxygen source. Usually that's the vasculature. Usually there needs to be some sort of blood vessels nearby for these cells to survive, and that's true everywhere. And so particularly for cancer cells, what ends up happening is that when there's insufficient blood flow for them to survive and want oxygen, they're going to release factors that induce angiogenesis. It's this idea of new blood vessels that grow out and facilitate tissue growth. This is important for tissue repair or tissue regeneration, but this is particularly important for cancer biology, where tumors will have these blood vessels growing into the tumor to provide access to nutrients that enable the tumor growth. The difference is that usually those blood vessels in and around a tumor are going to look a lot different than a healthy blood vessel. They're going to be more tortuous or more winding. They're going to be more leaky typically, but all enabling and trying to provide the nutrients to facilitate tumor growth. Whenever we talk about something being oncogenic, that means it's something that causes cancer. And in addition to that, you may have heard of this term before, but there's also genes that are typically associated with cancer. We call them oncogenes. What's important to note is that a lot of these genes typically have a normal function, but in cancer, they're mutated. So the mutated form of that gene, we typically refer to as an oncogene. The normal state of that gene, we typically refer to as a proto-oncogene. So proto-oncogenes and oncogenes are could both be referencing the same exact gene, but it depends on whether they're in their mutated state or their original state. If they're in their original state, that's a proto-oncogene. If they're in the mutated state, that refers to an oncogene. And we also have a family of genes that are meant to try and suppress tumor growth by nature of the function that they provide in cell biology, and we typically refer to those as tumor suppressor genes. And so there's going to be a host of proteins that we're going to be talking about that are tumor suppressor proteins, and the gene associated with them is identified as a tumor suppressor gene. And finally, we're also going to be talking about something we've referenced a few times throughout the class, but helpful to define. We're also going to be discussing growth factors. There are these family of proteins that typically, at a fundamental level, as the name implies, try and regulate and promote the growth, division, and maturation of cells. I'm going to step back and give space for any questions here. Yes. Always resolve issues. They're typically naturally there. So, for example, pancreatic cancer has a mutated form of RAS. So RAS we've talked about during both the MAP kinase signaling cascade when we talk about Noonan syndrome, as well as going back to our G protein lecture. RAS is a normal protein in cell biology. And so, however, when it's mutated, can actually lead to pancreatic cancer. So the mutated form of the RAS gene we refer to as an oncogene. The normal state of the RAS gene we would refer to as a proto-oncogene. Does that help make sense? Yes.
Not all diseases are cognitive.
Correct. They'll be associated with other diseases. So that would mean, for example, not every gene in the human genome is a proto-oncogene. There are only some that have been identified to be associated with cancer. So we refer to those mutated forms as the oncogene, and they're in the typically a lot of these proteins and genes are going to have touch on lots of different diseases. So when we're sitting in the context of cancer, we refer to their normal state as a proto-oncogene and their mutated state as the oncogene. However, it could be we might talk about them in a totally different context where we might not need to refer to their normal state as a proto-oncogene. Yeah. Yes.
Could you explain?
Yeah, so think of it as a degree of proximity. Invasion is usually talking about the ability of those tumor cells to move and migrate in kind of the surrounding area around the primary tumor. Metastasis is the spread of those tumor cells to a totally different tissue site and begin the growth of a totally different tumor there. So like typically when we talk about tumor metastasis, we think about secondary tumors that are emerging in a totally different tissue site. Like for the example with breast cancer, it'll typically metastasize oftentimes to either the brain or to into the bone. And so those other tissue sites we'd say are examples of metastasis. However, when you look at the primary tumor, the one that's in the original tissue, in this case breast cancer, for example, going back to this histology slide, we can see that it's invading to surrounding tissue. So that's an example of invasion where it's surrounding tissue around the primary tumor. Does that help make sense? All right, I'm going to press on. And so when we think about the incidence rates of some of these different kinds of cancer that emerge clinically, they typically will need time for a variety of these mutations to occur that lead to this cancer. And so there's constantly going to be exposure to different variables or factors that can induce some sort of mutation, whether that's UV radiation, for example, whether that's exposure to some other kinds of radiation, or ingesting different ingesting things that have been identified to have an association or a linkage to cancer that will induce some sort of mutation that over time can lead to cancer. And so what you can appreciate immediately is that a lot of these over time have a pretty similar slope, with the exception of being prostate cancer, where by nature of age, there's just an incidence rate of cancer that skyrockets as soon as you cross 50. But a lot of these other cancers have a pretty similar slope for their incidence rate over time on a logarithmic scale. And so what's important to note, if we think about like the evolutionary constraints around mutations and the protections around mutations, a lot of them don't really start to take off until age 30. And that's typically, I'm going to pick my words carefully. You can think of 30 or just north of 30 as a bit of a reproductive age limit, whereby there is a lot of protection to ensure survival of the species through reproduction. But after that, there's historically age, the association with survival was not much past 30. However, now with longer life, there's these ability for these mutations to accumulate that lead to higher incidence rates with higher age of different kinds of cancer. There are a lot of different kinds of genes or families of genes that are associated with cancer that are proto-oncogenes or can also be a tumor suppressor gene. And so the variety of growth factors or growth factor receptors. EGFR is one example, epidermal growth factor that's been closely associated with triple negative breast cancer. As an example, we have intracellular signaling proteins that have also been tied to cancer. One example we just talked about a moment ago is RAS. Transcription factors, in particular, have been closely associated with being proto-oncogenes that can, with a certain mutation, lead to or be associated with cancer. And lately, there have been a variety of cancer immunotherapies that try and address specific parts of these of this slide. Bless you. That, for example, try and target some of the cell cycle control proteins, checkpoint inhibitors, that have been a really successful cancer immunotherapy to try and heal cancer. And so, in addition to that, we also have DNA repair proteins where if there's some sort of mutation that's present in those DNA repair proteins or in cell cycle proteins as well, those mutations are going to have a much greater likelihood and have been identified to have a much greater likelihood leading to and being connected to cancer. So this was covered in the pre-lecture, but just to try and get our bearings, there's a few different stages in the cell cycle process. And so we're going to walk through it to try and connect some things that we've talked about throughout the semester that have a bearing on cell cycle here. And so starting at the top of the cycle, we have mitosis, where mitosis is the division of the nucleus, the separating of the chromosomes into subsequent daughter chromosomes. And so we talked about that during the actin cytoskeleton lecture, where we talked about how we have our tubulin that connects to the mTOC and the centromere of our chromosomes that begin to pull them apart. And so in the M phase, that's what's happening in mitosis. So we've talked about the molecular processes involved with that. When we think about that in the whole cell together, that's what's characterizing one part of the M phase, the mitosis part. Cytokinesis is the actual division of the entire cell itself. And so both of those are happening during the M phase, where now we get to the subsequent phases where another phase, the subsequent phases after that are referred to as interphase. And so the first one of them is the S phase. S stands for synthesis, where we have our separated chromatin, where now the DNA is going to be synthesized to try and produce a full chromosome for each chromosome now. And so the S phase stands for synthesis there. We have two other phases there that are G1 and G2. Those are referred to as just gap phases, where historically it wasn't quite clear what was happening during G1 and G2. And so they're just referred to as a gap phase because up to that point, only M phase and S phase were clearly identified for what was happening there. But there's what we've identified now is that there are clear functions of regulatory mechanisms at both G1 and G2 that try to ensure that what's happening during cell cycle is working properly, that there's no DNA damage, that there's no issues with any of the regulatory processes that try and make sure that cell cycle is occurring properly. And so in addition to that, we also have a G0 phase, which is this cul-de-sac out on the right coming off of G1, where we have this, where cells that are undergoing the state of quiescence or inactivity or cell cycle pausing, for lack of a better phrase, will go into G0 and become quiescent. That's distinct from senescent or senescence. Quiescence is this reversible state where they can go into a state of inactivity with respect to the cell cycle, but be able to enter back into it and undergo mitosis again. Whereas senescence is this permanent arrest that doesn't happen at G1, but typically happens at either G1 or G2. That's for senescence. Quiescence is this reversible pause in cell cycle at G0. That's quiescence. Senescence is this permanent, irreversible arrest, typically at either G1 or G2. I'll step back and give space for a couple of questions here. Yes. No, That's why the air quotes is it's not quite permanent. I meant to erase that because that's an old slide, but it is a reversible. I'll update the slide. G0 quiescence is a reversible state. Cells can become inactive and quiescent, but then in response to some activation signal, can become activated, grow, go through the cell cycle and continue moving on. Can eventually go back into G0 if they become quiescent again, but are able to come back out.
So it's like a kind of like you like put it on hold.
Yeah, you push pause on the cell cycle in quiescence. Senescence is a permanent stop. Yeah. Did I only post the slides? I apologize about that. I will post the video. Thank you for that. I appreciate it. Any other questions? Yeah.
Is it like developed beforehand when it's doing the computer, or is it developed?
So the connection with mutations, I want you to hold on to that because that's going to be what we talk about for the next few slides. The senescence G1 or G2. That's typically where senescence, that like permanent arrest typically occurs. All right. So when it comes to regulating the cell cycle, because this is such a fundamental part of cell biology, there are naturally going to be important regulatory steps that that feed into this. And so there's going to be a family of proteins that help regulate different aspects or different parts of the cell cycle. Those proteins are going to be cyclins. A second family of proteins that serve a regulatory role that might sound a little familiar to you guys are called checkpoint proteins. We have a few different checkpoint proteins that help stop the cycle in the case of some sort of damage, whether that's double-stranded break, whether that's some sort of mutation that alters the ability for replication to occur, whether there's some sort of mutation that alters the ability for spindle separation during mitosis. So a few of these places are going to be one at there's a G1 checkpoint protein that checks for very specific cues there. They're going to be primarily looking at cell size to try and cue up the cell after cytokinesis, checking for growth factors as well as DNA damage. There's going to be a checkpoint at G2 right before mitosis, right before the M phase. And so that's going to look for, again, assessing cell size to make sure it's prepared to undergo cytokinesis, and that it's also going to check for DNA damage to ensure that there's no double-stranded breaks that's going to create any room for mutations that might be perpetuated during mitosis. And finally, there's going to be checkpoint proteins at in the M phase, in mitosis, trying to make sure that there's appropriate chromosome attachment to the spindle, to those tubulin fibers. And so taking a step back to try and think about connecting a couple of these pieces together, when we think about receptor tyrosine growth factor receptors or growth factors and how they work, typically what ends up happening is that growth factors will bind to a growth factor receptor. Those receptors will typically, when they bind to a growth factor, whenever one protein binds to another, they each carry a charge with them. This goes back to lecture 1. When two proteins carrying their own charges come together, they'll induce some sort of conformational change. What ends up happening is that two of those receptors will come and dimerize together and associate together. And so once that happens, there's going to be the extracellular part of that receptor that binds to the ligand, but there's also a cytoplasmic part. For a lot of these growth factor receptors, it's not universal, but for a lot of them, the cytoplasmic part is actually going to be a tyrosine kinase. it's going to have the ability, the cytoplasmic tail has enzymatic activity. It's a kinase, which means that it phosphorylates. If it's a tyrosine kinase, that means it's specifically going to be phosphorylating tyrosine residues. What's unique about them is that when these two proteins dimerize together, there's going to be this phosphorylation on all the tyrosines on the cytoplasmic part that then will induce a conformational change and trigger activation of other proteins that lead to a cascade of events. For some growth factors, that cascade of events will include RAS, the monomeric G protein that we talked about a few lectures ago. And so again, we talked about how G proteins typically exist oftentimes as a heterotrimer. There are some that exist as a monomer. RAS exists as that exists in that monomeric state, and it contributes to the signaling downstream events that that we talked about in the RAS MAP kinase signaling cascade a few lectures ago. So to try and see what this looks like step by step, again, we have our growth factor receptor. In this case, it's epidermal growth factor receptor, or EGFR. It has an extracellular part, a transmembrane domain, and a cytoplasmic tail. There's a tyrosine kinase in that cytoplasmic part that phosphorylates tyrosines here on this tail. When it binds to EGF, its ligand, two of those receptors will dimerize together. There's going to be this autophosphorylation, where there's going to be a series of phosphorylation events by that tyrosine kinase domain on the tyrosines on that receptor. When those phosphorylations occur, phosphate groups carry a negative charge. When something carries a negative charge, it's going to change the conformation. Now that we have these phosphorylation events here on these tyrosine residues, that's going to enable signaling events that occur that lead to RAS activation. So we talked about how heterotrimeric G proteins are regulated. The way in which monomeric G protein RAS is regulated is going to be after we have the ligand bind to the receptors, and we have the dimerization of those receptors. Again, there's going to be the tyrosine kinase activity of that growth factor receptor. To get RAS, for RAS to be active, to be in its active form, what do you think it needs to be bound to? When we think about G proteins, primarily the alpha subunit, what is it bound to in its active state? GT. Yes, GTP. So it's bound to GDP. It needs to bind to GTP. The way in which that happens here is we have this other protein called SH2, Circ homology 2. There's a whole family of Circ proteins. Originally, Circ was identified by virologists, where there was this residue was found on viral. What was the story? It was originally found on viral oncogenes tied to carcinomas found on birds, but we identified that the same residues were found on these same important protein families. And so typically what ends up happening is that the SH2 domain binds to two phosphate groups, and it'll then activate. And this is it's part of the SH2 domain is part of a protein called GRAB2. The easy way to think about this is that it grabs two phosphates on the tyrosine kinase domain of this receptor. Once the SH2 domain of GRAB2 binds there, it'll then bind to this nucleotide exchange factor called GEF, and GEF now will is able to bind to RAS and exchange GDP for GTP. Okay, so we have again this activation of the growth factor receptor and its tyrosine kinase domain. We have a protein called GRAB2. SH2 is just like one part of that protein that we call it the SH2 domain. GRAB2 binds to GEF, this nucleotide exchange factor, that then helps swap out GDP for GTP. Now RAS is in its active form, and now that RAS is activated, it's able to trigger the downstream MAP kinase cascade that we talked about previously. Okay, and so with the activation of the MAP kinase cascade, one thing we didn't talk about is what MAP actually stands for. MAP stands for mitogen activated protein. Mitogen, meaning that it induces mitosis. So one of the key outcomes of the MAP kinase cascade is that it promotes cell growth, it promotes mitosis. And so this is going to be a critical pathway when it comes to cancer biology. And so MAP kinases, we talked about the dual specificity kinase where there's serine and threonine kinase activity. And so it's going to activate transcription factors that when they're phosphorylated, translocate into the nucleus and promote transcription and gene expression of specific genes. And so again, this connects back where we have activated RAS, where we have it activating RAF, which is a MAP kinase, MEK, which is a MAP kinase, which then activates MAP kinases. Again, mitogen activated proteins, they induce mitosis that then activate transcription factors. And in this pathway, in the context of cancer biology, what they're activating often, or what they're transcribing, is cell cycle regulators. So not only are they going to be inducing mitosis, they're also going to be regulating the expression of these regulators, these checkpoint proteins, and a second family called cyclins that we're about to talk about that contribute to the regulation of the cell cycle. These cyclin proteins have varying levels of expression, but will typically have specific species of cyclins that are present during G1, during S phase, and G2. And so in addition to the checkpoint proteins that are present at G1, at G2, and during the M phase, these cyclin proteins have varying degrees of concentration that is illustrated by these curves that as they get thicker, that corresponds to more expression of that specific cyclin that contribute to regulation of the cell cycle and driving G1 to S, S to G2, and then G2 to the M phase. And so the G1 cyclins, primarily cyclin E here, is going to help regulate the control of entering into the S phase. And similarly, cyclin A and cyclin B are going to be regulating the ability to go from S phase into, or G2 into the M phase. And so you can think of them, the checkpoint proteins are making sure everything is typically checking for DNA damage, cell size, the necessary nutrients to enable cell cycle. The cyclin proteins are facilitating entry and regulating different steps of the cell cycle here. Again, it's key to note that they vary in concentration, that there are specific cyclins for each phase, and during each of their respective phases, they vary in concentration over time, and that contributes to their regulation during specific aspects of each of these phases. We had more slides that go into more detail about each of these phases and specifically what these cyclins do. And there's a family of proteins that they each interact with that's marked on the previous slide called cyclin-dependent kinases. So the interaction of cyclins and those cyclin-dependent kinases have very specific roles in each phase. For the sake of time, was going to try and leave those out. But if you want to try and look into more detail about how cyclins interact with those kinases during each phase and the mechanisms of that regulation, that information is available to you. I'll give space for a couple of questions here. Yes. Yeah, so I'll post an announcement of the information I shared at the beginning of the lecture, as well as I'll also share specifically what slides I skipped. Yep. Any other questions? Yes.
Learning the cyclins.
Mm-hmm.
Is that like because of like some negative feedback loop, like from the S stage or like new stage, or
Sorry, you can go and finish your question.
right? Like, is there something that's like from the S stage that deteriorates I forget the actual name, the cyclin and G1?
Yeah, so each cyclin is going to be the expression of a given cyclin is going to be regulated by some transcription factor that's going to induce its expression. That pathway of how that transcription factor becomes activated is primarily dictating the regulation of those cyclins. So there's going to be activation of pathways during G1 that triggers cyclin E to be expressed, and that pathway will be turned off over time to stop expressing cyclin E. And then another pathway is going to be activated to express cyclin A and B. And over time, that pathway is going to be turned off. And so it's basically, you can think of it as turning the levers for one cyclin to be expressed, given what phase we're in of cell cycle, and turning it down if we're trying to be ready to transition into the next phase and express the next cyclin. Does that make sense? All right, we've got 9 minutes. I think I can get us there. So again, this is just to repeat that both cyclins and checkpoint proteins are critical regulators that provide feedback on cyclin enabling, going from one phase to the next, and checkpoint proteins serving as a bit of a regular, almost like physical checkpoint to ensure that there's no damage or no issues before entering to the next phase. And so these cyclins and checkpoints work together to make sure that whether a protein has any DNA damage or any issues, if it's prepared and ready to go into the next phase of the cell cycle. In the case of DNA damage or any issue emerging, we have mechanisms that try and address that. And so one of the most important DNA damage proteins in cell biology is a protein called P53. It is dubbed as the guardian of the genome. Typically, whenever a protein is named P and then some number, it was originally named before its function was known. And so P just means it's protein and the number is its molecular weight. So P53 has a molecular weight of 53 kilodaltons. But now we've come to understand that it has an incredibly central function in cell biology where it helps to provide protection against DNA damage. And so what ends up happening is that in normal, healthy cells, when there are no issues with the genome, P53 is marked with a tag called ubiquitin. It's been ubiquitinated, which means it needs to be degraded. So whenever you hear something has been ubiquitinated, that means it's marked for degradation. And so that typically means in a healthy cell, there's not much expression of P53 because it's typically being degraded given everything is fine. In the case where DNA damage is detected, there is activation of this pathway where P53, instead of being degraded, it's typically marked by MDM2 up here to be ubiquitinated for degradation. It undergoes a phosphorylation, and now phosphorylated P53 is actually a transcription factor itself that can now trigger the inhibition of cyclin-dependent kinase. That when you think about blocking some of these kinases that are connected to cyclins, if we inhibit cyclins, that's going to lead to cell cycle arrest. So P53 puts the brakes on cell growth when it's phosphorylated. It's going to lead to cell cycle arrest by nature of regulating a cyclin-dependent kinase. Now, what ends up happening is that if it stays phosphorylated, if there's a continual sensing of DNA damage, it's going to end up leading to apoptosis of that cell. It's going to undergo programmed cell death because of the extensive DNA damage that hasn't been repaired. So typically, again, P53 undergoes degradation when it's marked by when it's ubiquitinated. But in the case where DNA damage is detected, what ends up happening is that, excuse me, it's phosphorylated and it's a transcription factor and it transcribes a family of genes that ends up blocking cell cycle and enters into a state of cell cycle arrest. If the damage is extensive, it'll promote apoptosis of that cell. And so P53 mutations are one of the most common mutations in all of cancer because of this central role it serves in detecting DNA damage. And so whenever there's some sort of mutation that causes an issue in being able to regulate the sensing of DNA damage, because typically in cancer, there's some sort of, there's a variety of mutations that occur. Dysfunctional P53 no longer allows our cells to be able to, that cell to undergo cell cycle arrest or undergo apoptosis. A dysfunctional P53 in cancer allows that cell to grow despite detection of DNA damage. And the malignancy associated with cancer is typically not caused by a single mutation, but the accumulation of mutations is typically limited by P53. And so the mutation of P53 where it's dysfunctional opens up the door for more mutations to occur. And again, it's going to lead to more mutations that accumulate to more oncogenes. And there's many ways to lead to the dysfunction and alteration of P53. So bringing this back to HPV. HPV, the human papillomavirus, exists as a caspid that is composed of this protein called L1 that encapsulates the genome, the viral genome, the double-stranded DNA. And so if you crack open the HPV genome, you're going to see L1 encapsulating this double-stranded DNA. Typically, the cancer-inducing strains of HPV only have about 10 genes in their genome, as opposed to the human genome that's about 25,000. And so what makes HPV connected to cancer and what makes this so harmful is that during an HPV infection, that HPV virus is going to try and integrate its DNA into the host genome, into the host chromosome. And so it's the integration of the HPV genome, the HPV DNA into our DNA that makes those cancer-inducing strains so harmful. And so the 10 genes associated with the HPV genome are listed here. We could spend time walking through them. We're not going to do that today because that would be about a 20-minute exercise. But it'll be a good exercise for you to think about. It'll be a good exercise to kind of take all of the information we've discussed this entire semester to think about if there's this viral product in our genome that is encoded by that gene, how would that lead to some sort of infection, promote infection, or promote malignancy? That was bad timing. So we're not quite done. We're almost there. It's going to come back. Bless you. When the slide comes back, if cancer research is something you're interested in, we have a really strong presence of faculty here in the cancer space. Kristen Nagley specifically studies tyrosine phosphorylation. Kim Kelly has founded a company and dedicated her career to studying pancreatic cancer. And Kevin James has both studied virology and breast cancer and does really amazing work developing patient-specific organoids to study breast cancer. As I said, this is the last lecture. So I want to thank you guys for a great semester. I want to take a moment and thank the TAs. I don't know where Timothy's at. Oh, there you go. Sorry. You were over there earlier. So not all the TAs are here, but a lot of the work that goes into making this class operate is the TAs working behind the scenes to make more office hours available, getting the grading done. I'll totally have a moment of transparency. I was the weak link on getting the research statements back to you. The TAs did it in the appropriate time frame. And so they're the ones who are making everything work. And so I want to say thanks to the TAs. Finally, we didn't get a chance to do the journal club that I wanted to do, but to try and take a moment to identify that large language AI models like ChatGPT makes a lot of things really easy for you, but it doesn't think for you. It doesn't enable you to try and connect pieces together, to use the language of cell and molecular biology to understand studies, to determine, is this a correlative study or is this causal? And so what's important to know is that my hope for this semester is to equip you all to think like a cell and molecular biologist, to be able to look at not news articles, but to look at primary sources and to determine for yourselves, is there validity to their claim? Is there sufficient evidence to prove their claim? And so my hope is that you guys can understand how to read those primary sources, make a determination for yourself, learn how to ask compelling questions and find a way to promote, create a hypothesis from that, and for you guys to get plugged into research and answer some cool questions. So with that, I want to say thanks for a great semester. and hope you guys have a good summer.